Hello, my name is Etienne Sollier, and today I'm delighted to have the opportunity to share with you my work on joint copy number and mutation phylogeny from amplicon single-cell sequencing data. I'd like to start by telling you a little story about the bird, the bats, and the butterfly. As you can see, they can all fly, so you might expect that their most recent common ancestor already had wings. But surprisingly, this is not the case, and they all independently evolved the ability to fly. So it's an example of convergent evolution, and interestingly, by studying tumor phylogenies, we can show that the same phenomenon of convergent evolution can also occur in tumors. So I hope I've piqued your interest. Now I'll give you a little bit of background on tumor phylogenies. So let's say we start with a population of healthy cells. Then, one of those cells may acquire a somatic mutation, and if this mutation provides a fitness advantage, then after several cell divisions, most of the cells will have this mutation. Subsequently, other mutations can be acquired, and we can have patterns of branching evolution, where some cells will have some mutations, while other cells will have different mutations. And our goal here is to reconstruct such a phylogenetic tree of the somatic events that occurred in a tumor, because this can give us insights into how the tumor evolved and how it might resist against treatment. Several technologies can be used to infer tumor phylogenies, but today I will focus on targeted single-cell DNA sequencing, which is becoming very popular since it provides a very high throughput while still giving enough information to reconstruct phylogenies. Most of the targeted single-cell DNA sequencing data currently available comes from the Tapestry platform from Mission Bio. With this platform, we can sequence thousands of single cells, and for each single cell, we start from only one copy of the genome, so we have to start by amplifying the DNA. And here, only some amplicons are used, so typically we use panels which have between 50 and 300 amplicons, and which target genes of interest. Ideally, what we want is that if we have one heterozygous mutation, we want to find reads which have the mutation, as well as reads which don't have the mutation. But we can also have sequencing errors, where we find some mutations in some reads, even though there is no mutation in the actual tumor genome. And we can also have allelic dropouts, which is very common in single-cell sequencing. And this is when only one of the two alleles was amplified. So for example, here we might miss this yellow mutation. And these dropouts can potentially confound the inference, because if you look at the data for this cell, you only find the blue mutation and not the yellow one. So you might infer that the blue mutation occurred first, which is not the case in reality. Therefore, we need probabilistic models in order to account for these errors. Several methods have already been developed for single-cell DNA sequencing data. Most of them, like CITE, build phylogenies which are only based on single nucleotide variants. But this only provides a partial picture of tumor evolution because it ignores other important events like copy number alterations. BIT-SC2 can build phylogenies based on both SNVs and CNAs, but an important limitation is that it assumes that the coverage is uniform across the genome, which is not the case for targeted sequencing, and it's therefore not applicable to this type of data. So in order to bridge this gap here, we developed COMPASS, which stands for Copy Number on Mutation Phylogeny from amplicon single-cell sequencing data. With COMPASS, we considered four types of somatic events: SNVs, copy number loss, copy neutral loss of heterozygosity, and copy number gain. And as you can see, when a variant is present in a region, then the CNAs are allele specific. So with these four types of events, we can build phylogenetic trees. And here is one example where we start with one SNV, and then we have in one branch a copy neutral loss of heterozygosity, and in the second branch, another SNV, and a copy number gain of two regions. We also want to point out that our method can take into account germline SNPs, because this information can be leveraged in order to infer copy number alterations, for example here, this CNLOH. So this is one example tree, but what we want is the tree that best fits our data. So for this, we compute a likelihood for a tree, which tells us how well our data is explained by the tree, as well as a prior, which penalizes the complexity of the tree. So how many nodes and copy number iterations do we have in this tree? For the likelihood, we assume that the cells are independent, so we have this product here over all of the cells. But then, we do not know to which node in the tree each cell should be attached. Therefore, we marginalize over all of the possible nodes. And then, for each cell, we need to compute its likelihood given the genotype implied by a node. And for this likelihood, we consider two different parts. The first part consists in the allelic read counts at SNVs. So for each SNV and for each cell, we count the number of reads supporting the reference or the alternative allele. And these allelic read counts are modeled using a beta binomial distribution, but we also include dropouts and sequencing errors. Here is how this data, how this probability distribution looks like for several possible genotypes. In blue, if the cell does not have the mutation, we would expect the variant allele frequency to be close to 0. In yellow, if we have one copy of each allele, we would expect to find variant allele frequency close to 50%, but because of dropouts, we can have variant allele frequencies of close to 0 or to 1. And if we have a copy number gain, we can have an imbalanced variant allele frequency close to 2/3. As you can see, allelic read counts can already be used to infer copy number alterations, but the main source of information to infer CNAs will be total read counts in regions, which we model using a negative binomial distribution. The expected value for this distribution is proportional first to total read counts in the cell, as well as to the copy number CK of region K in this cell. So if we have a higher copy number, we would expect to find more reads covering this region. But there is one final parameter, rho k, which is a weight specific to each region. So as you can see here in real data, in targeted sequencing, each region is amplified differently, which is why we need this weight here specific to each region. With this, we can compute a score for each tree, but we still need to find the best tree. And since the tree space is very large, we cannot search it exhaustively, but we instead use a Markov chain Monte Carlo approach. So we start with one tree, and then we perform one small modification to this tree. For example, here we can merge these two nodes into one. Then we compute the new score for this new tree, and we either accept or reject this new tree based on this score. And this NC approach is performed twice. First, only with SNVs and without CNAs, because with the tree only based on SNVs, we can use the cells which are attached to the root, so which should be healthy diploid cells, in order to infer the region weights. And once we have inferred these region weights, we can run a second MCMC with both SNVs and CNAs. So this is our method. And then we evaluated it and simulated data and compared it against existing approaches. And here, for the performance, we use the tree similarity. So 1 means that the inferred tree is exactly the same as the correct one. In blue, you can see that site performs well when we don't have CNAs, but its performance drops when we have many CNAs. And this is expected since site only models SNVs, but not copy number alterations. Then in yellow, bit SC2 performs well when the coverage is uniform, which is what its model assumes, but its performance drops very sharply when the coverage is not uniform, so it's not applicable to single cell targeted data. Whereas Compass in red performs well in all of those settings. Therefore, Compass is the only method that can infer a joint phylogeny of SNVs and CNAs for targeted single cell DNA sequencing data. Next, we applied Compass to real data. So here I will show you data from two published datasets. and mainly from one large dataset of 123 acute myeloid leukemia samples. Here is one example tree that we can infer with our method. As you can see, the trees are rather small because with targeted sequencing, we don't cover so many somatic mutations. But there is still enough information to find interesting phylogenies. For example, here, COMPASS inferred two different subclones which had different copy number alterations. For this subclone, we also had bulk SNP array data, which can also be used to infer copy number alterations. And indeed, the deletion that we found on column 7 that was predicted by COMPASS was also found in bulk data, as well as the copy number gain on chromosome 11. However, the deletion in column 17 predicted by COMPASS was not found in bulk data, but this is expected since this deletion is predicted to only occur in 5% of the cells, so it cannot be detected by bulk data. So this example shows that COMPASS can detect clonal copy number alterations, which are in agreement with bulk data, as well as subclonal CNAs, which can only be detected by looking at a single cell resolution. In the introduction, I promised you some examples of convergent evolution, and here is already a simple one, where we have in two different subclones a mutation in IDH1 and in IDH2. So here, two different subclones independently acquired mutations which have a similar effect, so it's an example of convergent evolution. But since this example is only based on SNVs, this could already be found using existing approaches like SITE. But with our method, which incorporates both SNVs and CNAs, we can find new examples of convergent evolution. For example, in this myeloproliferative neoplasm, we found one mutation in EZH2 in one subclone, and in a different subclone, we found a deletion of the same gene. I'd like to finish with a more complex example of convergent evolution, which I find very interesting. In this sample, we start with two different mutations in TP53, and then we have four different subclones, two of which have deletions on chromosome 17, which affected different alleles. So here is one small sketch which explains what happened on chromosome 17. In the first subclone, the three genes targeted by our panel on chromosome 17 were deleted, whereas in the second subclone, only two out of three genes were lost. And in the two subclones, a different allele of p53 was deleted. When I first saw this sample, I was a little bit puzzled because usually deletions on chromosome 17 are selected for because they inactivate p53, whereas here, if both copies were already mutated, we would not expect a fitness advantage from losing one copy. But here, since this deletion was selected twice independently, it likely provides a fitness advantage. So here we looked in a large cohort of AML samples, and for each position on chromosome 17, we count the numbers of samples which have deletions at this position. As you can see, P53 is indeed the most commonly deleted region on chromosome 17 in AML, but there is also a secondary peak here on NF1. So NF1 is also frequently deleted in AML. So these deletions in chromosome 17 in this sample might have been selected for because of NF1. And this would actually make a lot of sense if you consider the two other subclones here with mutations in PTPN11 and KRAS, because they are all involved in the RAS pathway. So very briefly, RAS can switch between an inactive and an active state, and when RAS is active, it leads to cell proliferation. So the mutation in KRAS makes it constitutively active, which means that it cannot go back to the inactive state. And then the mutation in PTPN11 makes it more likely for rats to switch to the active state. And then where does NF1 stand here? Usually NF1 makes RAS switch from the active to the inactive state. But here, the loss of NF1 makes it more likely for RAS to remain in the active state. So in this sample, we have 4 different subclones which all activated the RAS pathway by a different mechanism. And again, this shows the importance of our method because this can only be detected if you look both at SNVs and copy number alterations. So to recap, here we developed COMPASS, which is a method for copy number and mutation phylogeny inference from amplicon single-cell sequencing data. And our methods can provide new insights. For example, here I focused on convergent evolution. And our method is available on GitHub, so you're free to try it out. With this, I'd like to thank everyone involved in the project, especially Katarina, Jack, and Nico. And if you're interested, you can read the full preprint on bioRxiv, and you can contact me. Thank you.